Here I am presenting a video about the various classes of the fungi. We study with the comparison of the two groups. They, this is a common class which is Phycomycetes. It is divided into two. One is Oomycetes, one is Zygomycetes. The Oomycetes, both are basically aquatic habitat. They have aquatic habitat. The Oomycetes and but still there are differences in between the two. We'll see what is that. The zygomyces is also called the conjugation fungi. Conjugation fungi. First, the step of division is what is the step component of cell wall. In oomyces, it has cellulose. Most of the fungi we see that there is chitin. But in case of oomyces, it is cellulose with pectin and other glucans. If we see the hyphae, both the hyphae of the Phycomycetes is cenocytic, that there is aseptate, they are multinucleated hyphae without any cell wall in between them. So they are aseptate and multinucleated in both Oomycetes and Zygomycetes. If we go to see the asexual reproduction in the Oomycetes is by the जैसे स्पोरेंजियोफोर बनता है उसके ऊपर जो स्पोर्स बनते हैं दे आर स्पोरेंजियोस्पोर्स दे आर ऑफ टू टाइप्स दे कैन बी मोटाइल लाइक एज ज़ूओस्पोर्स द ज़ूओस्पोर्स इन ओमाइसिस एंड विदाउट any flagella are aplanospores in zygomycetes. These zoospores which are having flagella, these flagella are, there are two flagella which are of different size, so they are heterocon and they are tessellated, they are hairs. So this type of the flagella we see in the spores, zoospores of the oomycetes. So asexual reproduction in oomycetes is by the zoospores and zygomycetes it is with the aplanospores. This is the first difference between those. Then there is difference of the cell wall. If we see the sexual reproduction, then sexual reproduction we are seeing here. In oomycetes, see there is this is an hypha with multinucleated hypha. On this hypha there origins on two different hypha there is the sex organs are appearing. These are because oogamous condition, the female will be large and round without any motile, and the male gamete would be motile and smaller in size. This condition called oogamous condition. This oogamous condition is present in oomycetes. So, and second thing you have to see that here the antheridia and archegonia are the two structures which are produced. There is gametangial contact between them, then that leading to the first somatogamy, then karyogamy, and then meiosis. Right. Here in case of the zygomycetes, it is gametangial copulation, that the cell wall in between them will disappear, and here it is not oogamous condition, it is isogamous condition. And the zygote which is produced will give rise to spores. After meiosis, they will give rise to spores. These spores are called zygospores. So the name of this is zygomycetes because zygospores are produced after meiosis of the zygote. So this was the difference between the sexual reproduction. Here it was gametangial contact, here it is gametangial copulation. Here it is oogamous condition, here it is isogamous condition. Here it is zygospores are produced in zygomyces. Then coming to the examples, here this is the examples are the late blight of potato that is caused by Phytophthora infestans, the white rust of cruciferous mustard caused by Albuva candidans, damping of disease caused by Pythium, the green ear disease caused by the Punicetum. You have to remember only three examples mainly, the Phytophthora infestans, Albuva candida and Pythium. Then here you see the examples are the Rhizopus, the bread mold and the mucor. The mucor also causes mucormycosis. So this was the difference between the oomyces and zygomyces. Here we are giving the second comparison is between the other two class, which is Ascomyces and the Basidiomyces. The Asco is called the sac fungi. This is called club fungi. Club fungi. The cell wall in both is made up of chitin. The hyphae in this are septate and they are branched. They can be branching. Septate and branched. The septate is having the pores, but the nucleus cannot go through these pores. Here it is again branched and septate, but the pores are different. Here the pores are called doliopores and they are clamp connections which are connecting one compartment to the other, and nucleus can move from one to another. In Ascomyces, the asexual reproduction takes place with the help of conidia. The conidia, but asexual reproduction is absent in Basidiomyces. Right? The motile structures are, matlab ki, there are no flagellated structures seen in the Ascomyces as well as in the Basidiomyces. If you see the sexual reproduction, the sexual reproduction is on the both the side it is with the plasmogamy. No special, this see, there are two hyphae. The plus means and minus means two different type of hyphae. You are denoting it with plus and minus, and there is fusion of the two hyphae, so it is called somatogamy. The first will be plasmogamy. Plasmogamy, there is the cytoplasm of the two will. Here these two nuclei have been fused with each other. Here two cells have fused and there are two nucleus. This is followed by making of an ascus. And these two nuclei will fuse karyogamy, and this karyogamy takes place and this karyogamy takes place within this ascus. After that, this zygote the nucleus will go meiosis to make the four ascospores. These ascospores are in the ascus. This fruiting body, here we have to see one more thing, that surrounding the ascus there are various mycelia. So it becomes a sac. The ascus is a sac, and there are 4 to 8 ascospores are produced in this asco. In this ascus there are 4 to 8 ascospores, and the fruiting body is called as ascocarp. While in Basidiomyces, when the fusion has taken the two nucleus, they have fused with the dikaryotic stage has appeared. There are two nucleus, and through this clamp junction, the nucleus will shift from one to another. So a special type of mycelium will produce which is having the dikaryotic in each In each cell there are two nuclei, that is dikaryotic stage of the mycelium. From this dikaryotic stage they will occur the appearance of the structure called basidium. Here then the two nuclei will shift and they will fuse. Karyogamy will occur in the basidium. This basidium, on the basidium, the four basidio spores will be produced exogenously. Here they were endogenous. Here these are seen, they are exogenous spores. The four basidio spores are produced on basidium and the fruiting body is called as basidiocarp. If you have seen as you see the example of the basidiocarp is the mushroom we eat. In mushroom we eat, what we see? There is a, in mushroom we eat, we see this is the shape of this mushroom. Here this is the stalk. On there mycelium. On the surface underground there are mycelium. The stalk is made up of the vertical hyphae. This is basidiocarp. The whole this is basidiocarp. These are gills, and under surface of gills, the gills are actually the basidium. On the gills there will be four basidiospores. Right. So here this is the fruiting body. The mushroom we eat is the basidiocarp, which is the fruiting body of the Basidiomycetes. Right. So here we learned about this sexual reproduction of the Ascomycetes and Basidiomycetes. Coming to the last group is Deuteromycetes. The Deuteromycetes, you have to remember, is the imperfect fungi. The asexual reproduction takes place with Cornelia. The mycelium is same, it is septate and branched as in Ascomycetes and Basidiomycetes, but sexual reproduction is absent. Right? If you see the examples, yes, examples of the Ascomycetes, as example of the Ascomycetes are the morels, the morcilla, truffles, right? Claviceps purpura, Erysipeus, penicillin, aspergillus causes aspergillosis. It's also the weed of laboratory. Wherever it grows, so it is weed of laboratory. Other is the Saccharomyces. The yeast is also an example of the Ascomyces. Saccharomyces, there are two types of yeast. One is used by bakery, which is Saccharomyces cerevisiae, and one is used in the production of wine, that is different, Saccharomyces elixodens. Then the gibberellins are produced from gibberella. This is also an Ascomyces. The neurospora is an Ascomyces. So these are the examples of the Ascomyces. Whereas the example of Basidiomycetes are what we smut fungi caused by Stelago, the mushrooms, the toadstools, the Puccinia. The Puccinia, you have to remember that in Puccinia it's caused a rust disease of the wheat, which has two hosts. I showed you in the video. It was barberry and the wheat. It was seen by K.C. Mehta. Other Basidiomyces are the puffballs, the bracket and the shelf fungi. Right. So this is about the examples of the Deuteromyces are left. Example of Deuteromyces are Coletotrichum falcatum causing red rot of the sugarcane, athlete foot caused by the trichophyton, the early blight of potato caused by Alternaria solani, Ticca disease caused by Sarcospora, and Trichoderma. Trichoderma causes a ring, also causing the infection. Trichoderma. Most of the decomposers of the litter are also the Deuteromyces. So here we revise this fungi. Thank you.